All right, welcome back to our second video for Chapter 7. Here we're going to look at what happens after glycolysis, including the oxidation of pyruvate, the citric acid cycle, and oxidative phosphorylation. So what happens after glycolysis really depends on if we have oxygen present or not. If we have oxygen available, and here we're going to focus on eukaryotic cells because we are eukaryotes, humans, then what happens next is the two pyruvate molecules we generated at the end of glycolysis will move into the mitochondria and become oxidized. And this process of oxidizing pyruvate creates something called acetyl-CoA that will be going into the citric acid cycle, the step after the oxidation of pyruvate. So during this process, let's look at what happens. I'm going to look at just one pyruvate molecule. I know I generate two from glycolysis, but let's look at one at a time. So the first step, it looks like, is I'm going to remove a carboxyl group from pyruvate, and that's going to be released in the form of carbon dioxide. It looks like when pyruvate is oxidized to acetyl-CoA, this is a redox reaction. So whenever something is oxidized, the other thing has to be reduced. Something has to be reduced because the electrons have to go somewhere. And it looks like it's NAD+ that becomes reduced and generates my high energy electron carrier, NADH. So if I look at the inputs and outputs, I have two pyruvate because I had two of them formed at the end of glycolysis. I need two of those NAD+ molecules, and I need two coenzyme A's. My outputs include two carbon dioxides. Each one is from each of those pyruvates, two NADH, and then two acetyl-CoAs that will enter the citric acid cycle next. So after the production of acetyl-CoA from the oxidation of pyruvate reactions, we move acetyl-CoA into the citric acid cycle. And again, just like in glycolysis, you do not have to memorize every single reaction that's happening here. We're going to look at and focus on what goes in, what comes out, where the reactions are happening, and notable characteristics. So if I look at the citric acid cycle, and by the way, this is often also known as the Krebs cycle, named after the guy who discovered this in the 1930s, I should say. And then sometimes it's also called the tricarboxylic acid cycle, although that name is pretty old. I don't really hear that as often. Most of the time people call this the citric acid cycle. The reason for this is because the first product of the cycle is citrate, also known as citric acid. So let's see what happens. In that very first step of the citric acid cycle, we take acetyl-CoA from the previous step, and that was two carbons. So each acetyl-CoA has the two carbons in it from the glucose. Remember, we had pyruvate, and we lost one of the carbons in the form of CO2 per pyruvate molecule. The acetyl is combined to a 4-carbon molecule called oxaloacetate. So this one has 4 carbons, this one has 2 carbons, to form citrate. Citrate is also a 6-carbon molecule. Then as we go through the whole citric acid cycle, we're going to be oxidizing citrate, and those 2 carbons we put in the form of acetyl-CoA will both be oxidized and released in the form of CO2. Along the way, it looks like we have some electron transfers, so some redox reactions, in order to generate 3 NAD and 1 FADH2 molecules. So we generate some of these high energy electron carriers because there are some redox reactions happening along the way. It looks like we also produce some GTP, which is synonymous with ATP, and which one we make really depends on which cell we're kind of looking at, what kind of cell we're looking at. And then as we go through the cycle, because we released 2 carbon dioxides, what we have left over is, again, oxaloacetate, the molecule we started with at the beginning. So this citric acid cycle keeps going on. It runs continuously because you're recycling your initial reactant. So the citric acid cycle only happens when we have oxygen available. And in eukaryotes, it happens in the mitochondria, specifically in the matrix of the mitochondria. Do you guys remember mitochondria are double membraned organelles? They have an outer phospholipid membrane, but they also have an inner phospholipid membrane that is folded up to increase surface area, and this inner membrane was called the cristae. The liquid inside, if I just put an X where this liquid is called the mitochondrial matrix, and this is where the citric acid cycle primarily happens. If I look at the pathway, if I just look at all of these reactions over here, even though the citric acid cycle requires oxygen, there's no direct use of oxygen here. So why do we only go through this pathway when we have oxygen? It's because all of these high energy electron carriers that we're producing, those 3 NADH and then that single FADH2, where's that third? There it is. 3 NADH and 1 FADH2, those are going to be shuttled to the next step where oxygen will be used directly. And let's see, I make only a little bit of ATP or GTP in this reaction, depending on the cell type I have. So there's not much ATP or GTP made directly. And again, there's no oxygen consumed directly. So let's tie this into everything that's happened so far. We had glycolysis, in which we started with glucose, a 6-carbon molecule. At the end of glycolysis, I'll just put gly, we had 2 3-carbon molecules. These were called pyruvate or pyruvic acid. And then I had that oxidation of pyruvate. So each of these pyruvates will get oxidized, forming acetyl-CoA, which is a 2-carbon molecule. Where did that 3rd carbon go? Remember, I lost it in the form of CO2. All right, so I breathe that out if I'm human. So I blow that out. I've got 2 carbons left. Each of these carbons, or 2-carbon molecules, enters the citric acid cycle. And interestingly enough, it looks like per turn of the citric acid cycle, which is per acetyl-CoA, I lose 2 more carbon dioxides. So by the end of the citric acid cycle, I have already oxidized all of the carbons from my 6-carbon glucose. All right, so let's look at all of the outputs so far from glycolysis, the oxidation of pyruvate, and the citric acid cycle. I know that I generated a net of 2 ATP from glycolysis, and then I saw 2 more from the citric acid cycle. Remember, we're going to consider GTP as equivalent to ATP for now. And the reason there's 2, even though I only saw 1 in that previous diagram of the citric acid cycle, I saw 1, is because, remember, there's 2 acetyl-CoAs, 2 2-carbon molecules, because of the 2 pyruvates that were generated from glycolysis. So I have a net of 4 ATP so far, at this point meaning at the end of the citric acid cycle. I have oxidized all of the carbons from glucose, as I just mentioned. And then I generated a bunch of these high energy electron carriers, NADH. Some of them from glycolysis, some of them from the oxidation of pyruvate, and many of them from citric acid cycle. I also made another type of high energy electron carrier, FADH2, also from the citric acid cycle. So as I mentioned, glucose is already completely oxidized by the end of the citric acid cycle, and I have a lot of potential energy stored in the form of these high energy electron carriers. So is most of the energy from glucose stored where? Is it in ATP, in CO2, these electron carrier molecules? We're going to say that it's in the electron carrier molecules. Remember, I called these the tickets from those arcade type games. So we're going to trade in our tickets for ATP in the very last step of cellular respiration. Our final pathway when we have oxygen available after the citric acid cycle is oxidative phosphorylation. And this is exciting. This is where we get to trade in our high energy electron carriers, our tickets, for our energy currency, ATP. So most of the ATP produced during cellular respiration occurs here. This is the only pathway where we use oxygen directly. And we're going to see that in this pathway, we have something called the electron transport chain, which I often abbreviate ETC for short, and chemiosmosis, which I mentioned earlier is the most common method by which we produce ATP. What we're going to see is the electron transport chain component of oxidative phosphorylation is going to create a proton concentration gradient. It's going to be an electrochemical gradient, and that is going to provide energy to power chemiosmosis, the production of ATP through chemiosmosis. And this is going to be happening in the inner membrane of the mitochondria. So we're going to see a couple of regions involved, but the proteins involved or the enzymes involved in this process will be embedded in the inner membrane of the mitochondria, the cristae. So let's look at that first part of oxidative phosphorylation, the electron transport chain, the ETC. Remember, we're looking at the mitochondria and that inner membrane of the mitochondria, the cristae. And these are where the proteins are embedded in that inner mitochondrial membrane. So this picture right here is basically here. Intermembrane space is the space between the two membranes, and the mitochondrial matrix is over here. So if I look carefully, it looks like what's happening is we have a bunch of electron transporters embedded in the membrane, and these are the blue proteins shown here. And we are going to be collecting electrons from those high energy electron carriers we produced earlier. So NADH will donate electrons to the electron transport chain in order to regenerate NAD+ for glycolysis and Krebs cycle or citric acid cycle. FADH will be doing the same thing. It's going to oxidize itself by donating electrons to this electron transport chain. As these proteins, these electron transporters, become reduced by receiving the electrons from these high energy electron carriers, they gain energy to push protons from the matrix into the intermembrane space. And as this happens, we're going to build up a nice proton gradient in this intermembrane space. Ultimately, we're going to let the protons back down into the matrix, which will be a very spontaneous or favorable process, releasing energy to form ATP. And we're going to see later that oxygen is required because the electrons that were donated to this electron transport chain have to go somewhere at the end, so there's no backup in the system. The final electron acceptor in the system is oxygen. So oxygen will receive the electrons that were donated from NADH and FADH2 to form water. So how do electrons move across the electron transport chain? What causes them to move? If we look carefully at the diagram here, there are four very large protein complexes embedded in that inner membrane of the mitochondria. And they are complex 1, 2, 3, and 4. Don't worry about what they're made of and all of the names of the enzymes. Just know that there are four complexes that are embedded in the inner membrane of the mitochondria. The reason electrons will be transferred from one complex to the next is because each subsequent component of the chain, of the electron transport chain, is more electronegative than the previous one. So it'll almost like suck the electrons to itself. Some of these components are mobile. They can actually move around. So one of these includes something called ubiquinone, or Q. Q can grab the electrons from complex 1 and complex 2, and transfer it to complex 3. Complex 3 cannot move, but we have something called, so that was ubiquinone, or Q. Complex 3 has something called cytochrome C that can also move around. So it'll take the electrons from complex 3 and transfer them to complex 4. Here, I told you earlier that the electrons have to go somewhere so that the chain is not backed up, and they go to the final electron acceptor, oxygen, which is, when it's reduced, forms water. So it'll pick up some protons as well to form water. If I look at the NADH and FADH2, they enter at different steps in the electron transport chain. NADH enters and donates its electrons to complex number 1, whereas FADH2 enters later on and donates its electrons to complex number 2. So the amount of ATP each of these high energy electron carriers can be traded for varies. NADH can be traded for more ATP compared to FADH2 because the number of ATP you can generate depends on the concentration gradient you generate. Since NADH donates its electrons to the first complex, what happens is when the complex receives the electron, it changes the shape of the protein, the complex, so that protons are pumped through, and several of these protons are pumped through. And then the electrons from NADH are going to keep going down this chain. Every time it goes down, more protons are pumped through into the intermembrane space. Whereas since FADH2, let me use a different color, FADH2 enters later on in the chain, it skips that first complex. So fewer protons are pumped through into the intermembrane space as a result of FADH2. So this is equivalent to fewer ATPs. Some textbooks will tell you that NADH can generate about 3 ATP. Others will say it's about 2.5. So between 2.5 and 3 ATP per NADH. For FADH2, some textbooks will say 2 ATP because they're rounding up. Others will say 1.5 ATP per FADH2 molecule. All right, so what's happening in the second part of oxidative phosphorylation is chemiosmosis, the more common method by which we produce ATP. So we generated that nice proton gradient where there are a lot more protons in the intermembrane space compared to fewer inside the mitochondrial matrix. And what wants to happen now is these protons really want to come back in because they want to go from high to low concentration. They cannot do so by crossing the membrane because these are charged. Remember, the hydrophobic layer, these fatty tails of the phospholipid bilayer, will not allow them to pass. So the only way they can get back in is through an enzyme called ATP synthase, this big blue protein here. As protons are allowed back in, going from high to low concentration, this is a favorable reaction. So that's going to be exergonic. It's going to release energy, and that energy is going to be used to power the endergonic reaction of the production of ATP. So again, this protein is critical for the production of ATP through this process, chemiosmosis. We're powering or using the electrochemical gradient generated from the electron transport chain to produce ATP. So if I look at the mitochondria, remember the inside is the matrix. Here we have our intermembrane space. This was the inner membrane. Here's my electron transport chain. And where did those NADH and FADH come from again? FADH2, I should say. Remember, most of them came from the citric acid cycle. Some of them came from that oxidation of pyruvate, and we also had some from glycolysis. They all eventually donate their electrons to the electron transport chain. ETC. We're using the energy from these electrons to produce our proton gradient, where there's going to be a lot more protons in the intermembrane space. And when we allow them to go back down, or with their gradient, that is exergonic. It releases energy, and we use that energy we couple that really with an endergonic reaction to form ATP. And again, the second half is called chemiosmosis. And this is just for fun, but there were two scientists that won the Nobel Prize in 1997 for the discovery of ATP synthase. And one of them was a professor at UCLA. When I was there, I remember I was there in 97, and we had our chemistry building. I forgot what the old name of the chemistry building used to be, but there was a big celebration for Paul Boyer. And after the Nobel Prize was awarded, we renamed the chemistry building to the Boyer Building. And now I believe several buildings at UCLA are named after him. So overall, if we consider all of the ATP that we produced through the method of cellular respiration, it's between approximately 30 to 36 ATP molecules per glucose. So this really varies depending on what kind of cell or species we're looking at. And it also depends on how efficiently NADH is shuttled from the cytoplasm of the cell. Remember, that's where glycolysis happens, into the mitochondria where the electron transport chain is found. Overall, we'll just remember somewhere in the 30s. That's how many ATP are produced per glucose molecule. Although we do lose some energy in the form of heat, cellular respiration overall is pretty efficient. We actually can capture about 34% of the oxidation of glucose that process in the form of ATP. And to compare this to like a car, probably not an electric car or a hybrid car, but a gasoline powered car, the efficiency of our cars is about 10% or sometimes less. So if you imagine you get like 10 gallons of gas and it's so expensive, only 1 gallon is actually powering the movement of your car. The other 9 gallons, that energy is lost in the form of heat. All right, that takes us to the end of our second video for chapter 7. In our third and final video, we're going to look at what happens if we don't have oxygen. What happens after glycolysis if there is no oxygen?